Hello and welcome to Building Your Best Brain podcast. My name is Sylvie, and I'm excited to dive into a saying that I've been saying a lot, and it's kind of been sweeping all over the internet for a while now, which is cells that fire together, wire together. With this sweeping the internet, I think it's super critical that, you know, it's a very valuable principle in neuroscience, but it's important to be able to understand what this principle actually means at the cellular level and also to kind of know the history, right? Like who thought up of this theory, how has it been tested, all that sort of stuff. So that's exactly what we'll get into today. And before we get into the history and the scientific evidence, there's a few things that are critical to know when it comes to the brain and circuitry in order to make sense of this saying. So we'll dive right in. So circuits are essentially neurons that are quote unquote wired together. And you'll understand why I use the quotations. There are other cell types in the brain too that of course play the role in circuitry, but we'll just stick with neurons for now. Neurons are unique that they're a really polarized cell. And what this means is that it's not a perfect circle. So on one end of a neuron, we have this branching known as dendrites. And these surround the cell body, right, which contains different organelles. It contains a nucleus with all the DNA, so the blueprint. And the dendrites are responsible for receiving information from other cells. And we'll get into what the information they're receiving is in a moment. And then you have a very long axon, which is a very kind of skinny, long extension from the cell body. It's wrapped in white matter known as myelin to kind of insulate, almost like a wire, right? The kind of rubber tubing around a wire. And then at the very end of the neuron is a terminal button. And so it's kind of just a little bulb at the end, and it's full of different neurotransmitters. So when I put in quotations that in a circuit, neurons are wired together because neurons are not actually physically touching each other. At the end of a terminal button, it'll create what we call a synapse with a dendrite, usually, of another neuron. And a synapse is a very, very small space. And this small space allows neurotransmitter release. So once a dendrite receives information, which is neurotransmitters that are binding to proteins called receptors, it'll change the electrical charge of that neuron. And once it reaches a certain threshold, this electrical charge will come all the way down the axon to the terminal button. And this is a all or nothing process. So it's not like you have weak action potentials or strong action potentials. Once you reach the threshold, the action potential is happening whether you like it or not. And once it reaches the end of the axon to the terminal button, this is what allows neurotransmitters to be released into the synaptic cleft. And something that's also worth noting is that not all neurotransmitters act the same, and that is because there are different neurotransmitters. There are catecholamines, right? So those are some of the common ones we've heard, right? So like dopamine, neuroepinephrine, or adrenaline. There's also others, so glutamate, GABA, and they also have various different receptors. So it's not one type of receptor per neuron transmitter. One neurotransmitter can have an insane amount of receptors. And they can either be excitatory or inhibitory. So this binding action can either change the charge so that that neuron that's receiving the information is more likely to fire, or it can change the charge in the way to go the other direction, to be less likely to fire. And so now this is where we come into what we call Hebbian learning. So Hebbian learning is what we associate with cells that fire together, wired together. And Hebbian learning is a term that comes from the work of a neuropsychologist called Dr. Donald Hebb. He's a Canadian from Nova Scotia, and he did this work at McGill University at the Montreal Neurological Institute in 1949. And he proposed a simple principle for how the brain wires together. So this wasn't through any scientific study. This was simply a theory that he proposed. And I'll read to you exactly what he wrote this theory to be. When an axon of cell A is near enough to excite a cell B and repeatedly or persistently takes part in the firing, some growth process or metabolic change takes place in one or both cells, such that A cell's efficiency as one of cells firing B is increased. And so what this suggests is that one neuron needs to fire repeatedly and consistently to take part of the action potential of the other cell. And that changes, once this happens, changes occur to make that firing more efficient. Cell A does not need to fire as much in order for cell B to have an action potential. Now, the famous rhyme came in quite a bit later. So it came in around 1992 from Dr. Carla Shatz, put this together based off of her work. And her work looked at the developing visual cortex in humans. And what she saw was that there was spontaneous yet synchronous firing in the visual cortex of developing embryos. Even though developing embryos, right, their eyes are not open, they're not taking in visual stimulation. And so based off this work, she kind of coined this rhyme that we've been using for many, many years after the fact. However, it was in the early 2000s from Dr. Abbott, who brought the causal neurophysiological link to Hebbian learning. And this group coined the term spike timing dependent plasticity. And so the work that they did was done through what we call, and the way this group studied it was through brain slices and electrophysiological recordings. A technique known as electrophysiology is typically through the use of brain slices. So you're able to slice a brain in different thicknesses and bathe it in kind of a pseudo cerebral spinal fluid. So our brain is in cerebral spinal fluid, which is kind of like a salt-like mixture to keep everything at a specific charge. And so you can have this brain slice kind of floating in the cerebral spinal fluid, still electrically active, and then you can take different probes to stimulate cells, and then you can take different pipettes and so on to be able to record the activity of those cells. I have a few friends who are doing their PhD using what we know as ePhys techniques, and it just blows my mind. It's very, very challenging technique to do. And so what Dr. Abbott's group did was they stimulated one cell and they played around with the time of stimulation and how many times they repeated the stimulation. And then they recorded the following cell, cell B, through these different timings. And what they found when they looked at excitatory neurons, when they stimulate other excitatory neurons, if cell A is stimulated 40 milliseconds or less prior to cell B, the synapse was likely going to potentiate an action potential. They play around with having longer lead times for stimulating cell A, and they found that the opposite is actually true, that those synapses can, what we call depress, so they can actually weaken and be less likely to fire. And so as Hebb predicted, all the way back in the 1950s, that this repeated stimulation and the timing of this stimulation is absolutely critical for the strengthening of synapses. And this is, synapse strengthening and synapse weakening all falls under the bucket of neuroplasticity. And so the ability of the brain to form and reorganize synapses essentially based off of many things, but in this case, neural activity. So I hope that you've potentially gained a new appreciation for this term, cells that fire together, wire together, as well as a solid understanding of what that term actually means at the level of your neural circuitry. And so as we go through different podcast episodes, we'll be using this principle to our advantage to be able to form new thoughts, to be able to form new habits. So stay tuned for that. In the show notes, I've included all the references, but please send me a DM on Instagram at synaptic Sylvie if you struggle to find any of them. And I hope you have a wonderful rest of your day. See you at the next podcast.